everyone, my name is Shivansh Gaur and I welcome you to my channel Biological. Today we are going to start with the topic codominance. It's a very important topic in genetics and you should pay a very close attention to today's lecture. Watch till the end and I will explain you what codominance is. But before that, I would also like to tell you that now I am there on the Unacademy and I am there teaching in the EAMCET category. So if you go to the Unacademy app, first of all download the Unacademy Learning app, and after downloading the Unacademy Learning app, you can find my live video lectures, special classes and everything there in the TS and AP EAMCET category. You can find my live video lectures there on the TS and AP EAMCET category. So download the Unacademy Learning app right now and find my video lectures there. I take video lectures on both zoology and botany. So if you're not there at the Unacademy Plus, you should definitely download the Unacademy app, learning app right now and become the Unacademy Plus subscriber. What the benefits will you get? You will get the benefit of the premium content at a very affordable price. You will get structured live courses and dedicated live doubt clearing sessions. You will get quizzes at regular intervals for self-analysis, and you will also get the different batches for the complete preparation in all the subjects, whether it is physics, chemistry, maths or physics, chemistry, bio. And there will be test series or DPPs, full length mock test, etc. with analysis, and this single subscription will give you this unlimited access. You will be getting all of this in just single subscription. Okay, there are more than 20 plus educators, 20 plus top educators there. There are 10 plus batches already running, 250 plus courses, 2000 plus lessons, and many more are added every day. You just use the code LIVESHIVANSH and you will get each and everything in just one subscription. So go for the Unacademy Plus subscription right now, because these batches, they are there already running for 2021 aspirants. You can go for the 12-month plan, which is 14,850, and you can also go for the 6-month plan. Use my code LIVESHIVANSH. You use my code LIVESHIVANSH and this code will give you additional 10% discount. So what else you can ask for? Go to the Unacademy Learning app, take the subscription, use this code LIVESHIVANSH. When you're buying the subscription, you will get additional 10% discount. See the batches that are going on: comprehensive batch for PCM and comprehensive batch for 2021 PCB. There are also crash course batches for the aspirants, for the 2020 aspirants. You know, there are crash course batches in which I am also there teaching with my biology crash course there. Okay, so the crash courses are there, with the rank booster batches, they are there. So enroll now. So now we will move on towards our today's topic, which is codominance. So see, when we talk about codominance, we first have to see that what is the basic difference in codominance and the complete dominance. There is complete dominance and there is incomplete dominance. Incomplete dominance. Okay, so I have already posted video on incomplete dominance and everything, so you can go and see my videos there. Today I am going to talk about codominance, but first of all, what is complete dominance? Complete dominance is in which the F1 it shows only one phenotype. The two phenotypes which are there in the parents, it shows only one phenotype, or only one phenotype is observed in the F1 generation. While in the incomplete dominance, the F1, it shows neither of the two phenotypes which are there in the parents. The phenotype is of the intermediate type. It resembles, it does not resemble, any of the parent. From both the parents, it does not resemble any of the parent. But in complete dominance, the offspring resembles the F1 generation. It resembles either one of the two parents. Now, what happens in codominance is the F1 the F1 resembles both the parents. It resembles the phenotype of the F1. It resembles both the parents. Okay, so in F1 you will observe both the phenotypes. How does it happen? Codominance, because both the alleles for the character, both the alleles, they are equally dominant over one another. For example, in complete dominance, as we have seen in, for example, see, plant height. If you take plant height, so you have, two differentiating characters, or the two contrasting characters. One is tall and the other one is dwarf or short. Now this this tall is completely dominant over this small t, this allele, and whenever this capital T and small t, they are together, only one is expressed, that is capital T is expressed, and the plant is tall. That is what happens in the F1 generation. While in the incomplete dominance, as we have seen in the dog flower, for example, if you take the example of dog flower, whenever the red flowers, they are capital R, and the white flowers are small r. But whenever there is capital R and small r together, which means in the heterozygous condition, this offspring, it does not resemble this parent, it is of the intermediate type. The phenotype is of the intermediate type. We get pink flowers. These are red flowers, these are white flowers. But the F1 which is observed, it is pink, so it does not resemble either of the two parents. Okay, but in codominance, what happens? We are going to take the example of blood groupism. In codominance. In codominance, we are going to take the example of blood groupism, that is ABO blood group system. ABO blood group system. Now, if you want to know what ABO blood grouping system is and what is RH grouping system is, then you can look at the videos, you can watch the videos. The link for the videos, they are given in the I button right here. You can click that and watch the video, on ABO blood grouping system. Now, in codominance, we'll take an example of blood groupism. See, in humans, we have ABO blood grouping system. We have ABO blood grouping system, and the gene which is responsible. Now, there are 4 types of blood groups. First of all, there are 4 types of blood groups: A, B, AB, and O. Okay, A, B, and O. Now, how are these blood groups decided? This is decided by a particular gene. A gene is responsible for deciding this particular group, blood groups. And how are these particular blood groups like? What is a specific condition in the blood which decides this particular, blood group A, B, and O? It is a presence of a sugar. It is a presence of a polymer sugar on the surface of RBC. On the surface of RBCs, these sugar, they protrude from on the surface of the RBC. Okay, so it is decided by a gene. It is decided by a gene, and the gene is called I gene. The gene is I, which is responsible for controlling this character. That is blood group. For controlling this character, this gene is responsible. Now, there are, generally there are 2 alternating forms of a gene. Generally we have 2 alternating forms of a gene, which is known as alleles. But in this case, we have 3 alternating forms. We have, for example, IA. We have the other alternating form called IB, and the third is small I. So these are the 3 alleles of this gene I, and this gene I is responsible for controlling the type of sugar which is present. So this I gene, if IA is there, if IB is there, and small I is there, so what happens when these alternating alleles, they are formed in combination, or they are present in combination? Now see, since there are 3 alleles, any 2 alleles can be present at a particular time in any individual, because we cannot have all the 3 alleles at the same time. We can only have only 2 alleles at a particular time. Now what happens is IA and IB, they are equally dominant over one another. They are equally dominant over one another. IA is responsible for, forming a sugar A, forming a sugar A on the surface of RBC. IB is responsible for forming B. Okay, and a small I does not code for any sugar. It does not form any sugar. It does not form any sugar. So it does not form any sugar. It forms sugar, sugar type A. These are just the just different forms of sugar which are produced at the surface of RBC. Okay, so now what happens is this A and B, these 2 alleles, they are equally dominant over one another. So what happens is whenever these 2 alleles, they are present together, whenever these 2 alleles IA and IB, these are present together, it results in the presence of both the type of sugars, that is A and B, on the surface of RBC, and the resulting blood group of an individual becomes AB. The resulting blood group of an individual, it becomes AB. Okay, but this IA is completely dominant over small I because it does not produce any sugar, and this IB is also completely dominant. It is completely dominant over small I. So whenever IA is present with this small I, it is completely dominant over small I, and only this is expressed. Only IA is expressed, and it results in a blood group A. It results in a blood group A. This will result in a blood group AB, and this results in a blood group A. Same way, since IB is completely dominant over small I, the IB and small I combination will result in a blood group B. Okay, now one more combination is possible, and what is that combination? The other combination could be that both the alleles are small I, isn't it? This is also a possibility that both the alleles can be small I. In this case, no sugar is present on the surface of RBC, no sugar is there, and the blood group becomes O. So these are the conditions which are possible. These are the possible conditions which can be there. Okay, so see, what we are watching here is that A and B, they are equally dominant over one another. Okay, so no one is completely dominant. There is no incomplete dominance. There is equal dominance among the alleles, and this results in codominance. This results in codominance because now it will resemble the phenotype. The phenotype will resemble both the parents in equal proportion. So A sugar is also there, B sugar is also there. Since there are 3 alleles, as you can see, there are 6 possible genotypes. Okay, since there are 3 alleles for this particular one character. There are 3 alleles. There are 6 possible genotypes. And what are those possible genotypes? Let us now make a table, like a small table. Let us now make a small table and try to understand all the genotypes and their phenotypes. So now we'll see that since only 2 alleles can be present at a time in any offspring or any individual, so we'll see all the combination, all the genotypes, as well as the type of blood groups. We'll see that. Now let us see. Let us suppose this is allele from parent 1. This is allele from parent 1. This is allele from parent 2. Okay, this is allele from parent 2. Okay, then there is genotype, and there is blood group. What will be the blood group? We'll see that one by one. Now see, just in case, the first case, the allele from the parent 1 is IA, and the allele from parent 2 is small I. The genotype will become IA, small I, and the blood group will be A. Okay, since IA is completely dominant over small I does not produce any sugar, therefore the blood group here produced here will be A. Okay, now see, second case Second case is, from the parent 1 you have I, for example IA, and the second parent, from the second parent you have IB. You have IB. Now since IA and IB, they are both equally dominant over one another, first of all the genotype will become IA and IB, and the blood group will be AB. Any problem? No. Okay. Now the third case would be from the first parent you get IA, and from the second parent as well you get IA. This could also be the case that from both the parents you get IA. Now since both the alleles they are A, so the blood group will be, the genotype will be IA and the blood group will be A. All the three cases clear? Okay. Now moving further, fourth case. Now what could be the another case? From parent 1 you get IB. From parent 1 you get IB, and from parent 2 you get small I. The genotype will become IB and small I. The blood group will become B. The blood group will become B. Okay. Now the fifth case. What is the fifth case? From the parent 1 you get IB. Okay. And from parent 1 you get IA. The genotype becomes IA, IB, and the blood group is again AB. Clear? Sixth, from parent 1 you get IB, and from parent 2 as well you get IB. Okay, and the genotype becomes IB, and now the blood group will become B. Now the another last but not the least possibility is that from parent 1 you get a small I, from parent 2 as well you get small I, and the blood group becomes, the genotype becomes a small I, and the blood group will be O. The blood group will become O. Okay, now see, since these two genotypes are same, see here, these two genotypes, they are same, so they will be counted as one only. Since these two genotypes are same, the blood group is also same, they will be counted as one. So since they are counted as one, the possible number of genotypes are 6. We have seen 7 cases, but the total number of genotypes, different genotypes which are possible is only 6. Because these two genotypes they are same, so they are counted as one only. Okay, so now see, in these 7 cases there are 6 different types of genotypes. So since there are 3 alleles, there are 6 different genotypes they are present. Okay, this is also a very good example of multiple allelism. What does multiple allelism means? When more than two alleles they are responsible for controlling one character. When more than 2 alleles they are responsible for for causing the different phenotypes in a particular character, then it is known as multiple allelism. So this blood group system, this ABO blood group system, is a good example of a multiple allelism. Now see, since only 2 alleles can be present at a particular time, this multiple allelism cannot be observed in a single individual, because in a single individual you will only get 2 alleles. So this multiple allelism, you cannot observe this multiple allelism occurring in a single individual. You observe this multiple allelism only when you do studies at a population level. So when you study population, you experience or you see the multiple allelism, that is more than 2 alleles they are responsible for controlling one character or one the different traits of a particular character. Okay, so I think this is clear to all of you. The as I already told you when I was discussing the blood groups, the different blood groups in ABO blood grouping system, I told you that I'll shortly make a video on the genetics of the blood grouping system also, and today we have done that. And this is codominance. We have discussed codominance. Okay, and now very shortly I'll also be making one more video on pleiotropy as well, as I'm getting a lot of demand to make a video on pleiotropy. So stay tuned, Subscribe my channel, follow me on Unacademy as well. Follow me on Unacademy as well, because there you will be getting my live lectures for the EAMCET. And if you are preparing for NEET as well, then too you can come and take my lectures, because I'll be covering the syllabus of EAMCET, and of course the NEET is also covered in that. So if you're preparing for NEET, you can come. If you're preparing for EAMCET, you can come and take my live lectures on the Unacademy learning app. So do follow me there. Till then, subscribe my channel, press the bell icon so that you get the notification whenever I post a new video. Till then, take care, bye.